thank you to Precision NanoSystems for bringing us today's webinar. It is my pleasure to introduce today's speaker, Dr. Anitha Thomas. Dr. Thomas is the Director of Delivery, Research, and Development of Precision NanoSystems. She completed a PhD in medicinal chemistry from the Indian Institute of Science. Dr. Thomas joined Precision NanoSystems in 2013. She's an expert on the next generation approaches for nanomedicine development in the areas of vaccines, non-viral cell-based therapies, and genomic medicines, and leading projects at various stages of drug development. Dr. Thomas holds several patents in the field of LNP-mediated nucleic acids delivery and currently leads delivery program efforts at Precision NanoSystems. Dr. Thomas, the floor is yours.
Hi everyone. Welcome and greetings. Am I presenting in the presenter mode? I think everyone can see me. Thank you, Stacy and Ryan, for the introduction. So today, I will talk to you about novel ionizable lipid library to enable the non-viral delivery. In this webinar, okay, one second. Yeah. So this webinar describes novel ionizable lipids used as a functional excipient for designing vehicles for nucleic acid therapeutics, advancing targeted delivery where treatment options are absent. So through this webinar, we could talk about leveraging a library of novel ionizable lipids to design potential therapeutics, identify, optimize, and scale up potential LNP compositions using microfluidic platform technologies, review exemplary data sets showcasing various emerging applications. So as an outline, I would be talking about the need of the ionizable lipids in this phase. And then I'll walk you through the various proof of concepts in the space of cell therapy, vaccine applications, gene editing, and protein replacement applications that we have showcased. And in the end, I will actually talk to you about the challenges in the nanomedicine development, especially as it applies to the lipid nanoparticles, and how precision nanosystems can help with the scaling up and lipid nanoparticle manufacturing technology. So I will also talk to you about the plug and play nature of this technology, and this is what I was hoping to achieve through this webinar. So genetic medicines means nucleic acid plus its delivery systems. RNA and DNA are large molecules that require the nanoparticle delivery technologies to protect the nucleic acids from endonucleases and exonucleases and get into tissues and cells. So in this pictorial representation, RNA or DNA is the active pharmaceutical ingredient, and the synthetic lipids or polymers are the excipients. Together, they form the drug product. And as you may be aware of it, right? Like because mRNA vaccines have become the topic of discussion actually these years. And we need controlled self-assembly of the LNP drug product. Multiple components must self-assemble in an optimized manner to produce the desired drug product. So genomic medicines with lipid nanoparticle delivery. Lipid nanoparticles have become the leading delivery systems to transport nucleic acids into into the in vivo or ex vivo situations. Ex vivo situations haven't explored that much, but in vivo, you can use any routes of delivery for in vivo applications. And ex vivo, in an ex vivo applications, patient cells, like for example, red blood cells are you can take a blood draw and then isolate the T cells and then expand the T cells and transduce them. Currently, the viral delivery systems are being used to that purpose and then put back into the patient. And for all of these things, the mechanism of uptake is an endocytosis-based mechanism, where LNPs are actually like trafficked by the same mechanism as the cholesterol-mediated mechanism. And in the cytoplasm, an early endosomal release happens when the pH is actually being changed. So to come back to the topic of ionizable lipids, actually, this is actually probably the second largest subject that we have heard in the pandemic, mRNA vaccines and lipid nanoparticle. So we need more ionizable lipids to enable the genetic medicines. So, and why are we using the lipid-based nanoparticles? Because they are clinically validated. So LNPs have been clinically validated to successfully deliver RNA. And in 2018, the siRNA LNP on Patro has come out. And with that, actually, that has given the momentum to develop quickly pivot technology to develop mRNA lipid-based nanoparticles. And two approved drugs now, Comirnaty by BioNTech Pfizer and Spikevax by Moderna. So mRNA vaccines have been the largest proof of concept that we have ever achieved using for lipid nanoparticles. And I could only see that this will give a momentum for continuation of the lipid-based nanoparticles, enabling genetic medicines. And today we talk more about the ionizable lipids that are needed. So in general, like most of you are familiar with the structures of ionizable lipids and what are the top ionizable lipids that are clinical that have shown activity clinically. So most of the ionizable lipids, they come in different shapes, but there is a polar region and there is a non-polar hydrophobic region. The polar region is referred to as head and the non-polar region is the tail. And this is the structure of MC3 that's in the on-patro drug by Alnylam. And then we have ALC0315 in the Pfizer BioNTech vaccines. So you could see actually that now the newer lipids have branched chains from a central carbon atom. And then there is also like SM102. Again, you could see the branched chains, and there are also biodegradable ester groups in the newer forms of the lipids compared to the MC3-DMA, where the functional group is actually near the head group. So this is another lipid that's people have widely utilized. It's called a CTOL-200. Again, there is actually multiple tails. So all of these features actually are quite important when it comes to ionizable lipids. They affect actually how cells are, how the lipid nanoparticles are being uptaken into the cells, and how they are trafficked inside, and how the endosomal release happens. So there is a lot of interactions between the lipid components. So ionizable lipid is one of the main constituents of the lipid nanoparticle, and the head group actually affects the pKa of the ionizable group. pKa meaning that pK is the pH at which 50% of the ionizable group is ionized. So that means there is 50% in the positively charged form and 50% in the neutral form. So there has been significant literature talking about the pKa and how it influences. I wouldn't be going into the detail, but these are the general terminologies that need to be known in the background, such that to understand how these are being played. Now, again, So I would also just kind of actually give you like an idea about how these the 3D projections using ChemDraw, how the SM102 look like, and how ALC0315 looks like, just to have an understanding about how in a three-dimensional way it looks, and think about actually that the structures also affect the self-assembly of the nanoparticle. Then again, like I would like to show the MC3-DMA because which is in the ONPATRO, the first approved drug with a small interference RNA, and the CTOL200, another widely used lipids. So ionizable lipids are really needed for nucleic acid medicines, which are the next generation genetic medicines when it talks about non-viral delivery. As you can see, actually, there are only 3 lipids that are actually have a clinically demonstrated knowledge. Biopharma needs more lipids, ionizable lipids, to accelerate the genetic medicines. So Precision have this mission to accelerate the transformative nanomedicines. And we have been earlier actually working towards decreasing the formulation barrier for lipid nanoparticles. And then now our customers were interested in getting the ionizable lipids because of the access, the limited access of ionizable lipids. Hence, we went forward with a synthesis of ionizable lipids for enabling the genetic medicines. So we have something called the GenVoy platform, which is our reagent platform. Most of you may be familiar with the name GenVoy. We also have GenVoy ILM mix, so that's for general formulation development. It's more like a show and tell lipid formulation for people to enable and feel how nanoparticles can be made and manufactured using the microfluidics based mixing platform. So we have mentioned already that ionizable lipid is just only one among the main constituents of the lipid nanoparticle. The other constituents such as cholesterol, helper lipid, and stabilizer, they all play important roles in making the nanoparticles. So ionizable lipid, just to kind of remind everyone, it binds and protects the RNA, it facilitates the endosomal escape and neutral charge at pH 7.4. I forgot to mention here, like most of the ionizable lipids that have activity are in the range of like 6 to 6.7 range. And this is about experimental pKa that I'm referring to, and which facilitates the endosomal escape, and that also gives more safety profile because these are actually completely different systems compared to the earlier cationic lipids of the like 20 years back. So when the ionizable lipids are needed for actually encapsulating the nucleic acids, and then that's at a lower pH, and then when the charge is neutral in a biological milieu, it will actually becomes neutral in charge at pH 7.4. So the cholesterol is important for the endocytosis via the LDL receptor and ApoE mediated uptake, and the helper lipids gives the structural influence for the lipid nanoparticles, and the stabilizer, it stabilizes the particle by preventing the aggregation. It also protects from opsonization, and there are a couple of other influences also from the PEG lipids. So now I have mentioned actually that we have developed novel ionizable lipids to make access to the ionizable lipids for wider biopharma, and they are very proprietary state-of-the-art lipid nanoparticle technology and associated intellectual property. And also this is ionizable lipid is one, and then it's also about the total composition that we have to be taken into account, which is the LNP So we have taken an approach for specific relevant application-based screening hits for cell therapy, vaccine, and protein replacement and gene editing models, because PK is just only one parameter. There are lots of other parameters that we have to take into account, and also the biological scenario is important for it with respect to the target product profile nature of it. So we do have a diverse library of novel ionizable lipids, and from there, the lead candidate can be selected based on the applications that needed to be employed in there, and then the novel compositions can be designed further from the specific applications, and that kind of an approach we would like to explore. So in the following slides, I will walk you through the relevant applications that we have discussed in here. And first, I would like to talk to you about the vaccine application, because this is pandemic response time. Everyone has pursued the vaccine applications, and so are we actually. And we have got Canadian government funding to develop the infrastructure for vaccine development and become a pandemic response center in case if Canada needs to have. So we have used, instead of the mRNA, we have used another technology for vaccine application, which is called the self-amplifying RNA. So in many ways, actually, mRNA and self-amplifying RNA is similar in terms of manufacturing. So there is a self-amplifying RNA, and once it is in the once you have intramuscularly administered within the cells, self-amplifying RNA molecules actually they multiply, and more RNAs will be generated, which produces actually more antigen. As a result of this, actually, self-amplifying RNA has the potential to allow for dose pairing, and there is also the chance that actually a small amount of, or a minute quantity of self-amplifying RNA will be as potent as that of much higher quantity of mRNA. So the advantage of the mRNA and self-amplifying RNA, everyone knows about it. The antigens will be produced within the body without producing the disease-specific burden. So, and then it produces the antibody, the body produces the antibody-potent immune response. And so here is an example we have used using our off-the-shelf Genvoy ILM for effective delivery of mRNA and self-amplifying RNA. Here we showcase a luciferase protein expression. This is just to get you an idea about how 1 microgram a self-amplifying RNA is almost equivalent to 5 microgram of mRNA in terms of the expression. So when you look into the left side, you could see actually the profile of the mRNA, 5 microgram administered intramuscularly. And you could see that at 6 hours, it actually increases in the A expression. But when it comes to self-amplifying RNA, you could see that the onset of the expression differs, and almost day 6 onwards, actually, the expression continues. And unfortunately, we haven't explored further because it was one of our first experiments, actually. And we thought, okay, by 9 day, we should cover all the region. But when you look at the time response, you could see that actually the mRNA and the self-amplifying RNA follows different curves, different profiles. So again, there is difference between the administration, like a 1 microgram and 5 microgram dose, not compared at the same quantity. And in this case, actually, we have also compared the Genvo ILM with the MC3, and you could see that it follows almost the same profile, actually. So this is an example from our vaccine program. This is a study in hamsters following a SARS-CoV-2 challenge. So we had 2 vaccine candidates, and B and C in here, or we can mention it as 1 and 2 in here. And you would see that the vaccine candidates have protected the hamsters from weight loss. And the untreated animals had a huge weight loss. And in the second graph, you could see actually that the vaccine candidates prevented from the increased lung weight compared to the untreated control. In here, you could see how anti-spike titers, which is SARS-CoV-2 specific IgG antibodies, and then they're neutralizing the antibody titers for both the vaccine candidates. Both the vaccine candidates are similar in activity. And in the we have also done the nasal wash and the tissue wash, and vaccinated hamsters had a lower SARS-CoV-2 viral loads in both nasal wash and respiratory tissues. So it's just to give us an idea about the size and PDI in here. Their usual range is 70 to 80 nanometers in size with an encapsulation efficiency, with a very high encapsulation efficiency. So in summary, actually, vaccine candidates made with the proprietary PNI ionizable lipids using the NxGen microfluidic platform protected the hamsters from this challenge, from the SARS-CoV-2 challenge study. So now that we know that the vaccine candidates are efficacious, what we have done is actually we wanted to understand the in vivo potency of this self-amplifying RNA LNP for a long time, over 12 months, and in a biological situation. So we have administered the vaccine in day 0 and then followed through day 21. And each time a vial is pulled out and administered into the animals. So, and then we have followed this, one particular batch for 12 months. So you could, it's really surprising to see actually that it had a stability of 4 degrees Celsius for 1 month and minus 20 degrees Celsius for 2 months. And then the main objective was of the study was to actually look at 12 months at minus 80 degrees Celsius. And, we could see that the SARS-CoV-2 spike-specific IgG in the mouse serum remains the same. So it just shows actually that self-amplifying RNA LNPs are stable for 12 months at minus 80 degrees Celsius. So after getting the mouse, rat, and I have not shown you the rat study, but and hamsters data, we thought actually we wanted to see how whether these our ionizable lipids comprising LNP with a self-amplifying RNA payload, whether it will actually show efficacy in non-human primates. So we have used rhesus as the monkey here, and we have followed the prime and boost regimen, and in this case we have used an 8 weeks boost interval. And this is, we have only done 5 microgram of self-amplifying RNA. If some of you are remembering it, the previous, the mRNA vaccine developers have actually showcased the NHP data at a very high dose. So in the right graph, you could see actually all these vaccine candidates that we have tried is actually exerting more or less actually same activity and day 42, following the prime, there was really good activity. And the day 70, following the boost, there was really good activity. So the response on day 70 is much higher than that the other time points, which is a really typical response to the boost. And again, size and PDI is normal range, 60 to 85 nanometer difference. So in this study, actually, we have looked at different objectives. One was actually to understand the, antigenic response using the same lipids. And the one was to understand actually the lipids difference using the same antigen. But in all, like we could see actually really good response from the vaccine candidates that we have explored. So now, so that was about like one or two lipids that we have showcased before with, from our lipid library. So herein actually we wanted to screen some of our new lipids and against the clinically relevant lipids. So in this study, you could see that we have used biopsy mice and followed a prime and boost regimen of 4 weeks with 1 microgram of self-amplifying RNA, and showcased actually activity of some of our lipids against ALC0315. So the data indicates that the PNI lipids are extremely effective for vaccine application, very similar to the clinically relevant lipids. So this is another screen where we look at more lipids that has come out of our platform and have evaluated them against SM102. And again, the same approach, IgG levels following the prime and boost of 4 intervals with the 1 microgram dose. The data indicates that a number of PNI lipids are extremely effective for vaccine applications. So, in summary, like we could say that actually we do have lipids that's available for our vaccine application. If customers are looking forward to getting a particular vaccine application done, we would have a really good choice to suggest for them such that they will be successful. So till then we have talked about the vaccine applications using the novel ionizable lipids. Now we would like to talk about actually some protein replacement applications. So in this particular model, we have used anemia as a indication. So in a natural process of anemia, the kidney produces kidney produces apoprotein more, and the bone marrow stimulates the red blood cell production, and the correction of anemia happens. So in the current treatment regime, erythropoietin protein is used as epoetin alpha is one of the brand names, and it is administered by IV, and the bone marrow it stimulates the production of red blood cells. But when it comes to a gene therapy setting using lipid-based nanoparticles, one could actually give erythropoietin encoded mRNA, and the body produces the apoprotein by itself, and then the bone marrow could stimulate the red blood cells, production of red blood cells, and the correction of anemia can happen. So earlier, using the novel ionizable lipids, we talked about vaccine applications. At that time, we have used a self-amplifying RNA, but in this case, we are talking about mRNA. So once we have established an animal model for a specific applications, we continuously screen our lipids and identify the top leads for that particular application to enable our customers and accelerate their medicines to clinic. So in this example, we have screened a lot of lipids against with the erythropoietin encoded mRNA, and we have looked at the EPO levels, erythropoietin levels in mouse serum at 6 hours. Using a dose of 0.5 milligrams per kilogram at 6 hours terminal bleed. Again, the data indicates that PNR lipids demonstrates its ability to deliver mRNA encoded for therapeutically relevant proteins. And here, in this case, we have used the MC3 as a benchmark. So I would now want to talk to you about the gene editing applications using novel ionizable lipids and CRISPR and other gene editing. I wouldn't be talking to you about the biology of CRISPR or gene editing much. We believe that our customers are actually more experts than us in this regard with respect to the biology and the drug substance here, which is gRNA and mRNA. So we have used Cas9 mRNA in some cases, and then I will also talk to you about the other editing. So we have used the targets TTR and PCSK9 for this to showcase the gene editing application. So gene editing can be a transformational therapy which offers permanent cure. So, there is a lot of promise out there about gene editing, and with LNPs being very popular as a drug delivery system of choice, gene editing applications should be explored and should be actually brought it into the community. There are a lot of rare diseases that are waiting for finding a cure, right? With the advent of the genetic medicines, every target now become druggable. That was the main difference from a small molecule or a biologicals to the genetic medicines front. And so with that respect, actually, there is a lot of targets that can be explored, and a lot of diseases are waiting to be cured. So in a recent publication, we have looked at the genes with the reported evidence of gene editing efficacy in large animal preclinical studies or in clinical trials. And these, the genes actually that's been identified, the PCSK9, ANGPTL3, TTR, DMT, KLKB11. So these, most of these genes have one or the other way connected to cardiovascular diseases, which is kind of like one of the major diseases of the humankind, right? So these genes are really complicating and most often cardiovascular disorders happens as a result, and we have chosen PCSK9 and TTR as the targets, and I will also show you some exciting things that's happening in the field. So one is CRISPR to reduce the transthyretin amyloidosis, and Intellia's NTLA 2001 phase 1 clinical trial data. It's very exciting, and it shows high promise for transthyretin amyloidosis as a single dose treatment. This is the clinical trial number, and it's a really exciting time to be in the industry, and we look forward to seeing the positive results of this. Actually, it's a really good time. When it comes to the transthyretin amyloidosis, it's a life-threatening genetic disease, and it's caused by the accumulation of misfolded proteins, and it gets accumulated in the tissues of nerves, heart, kidneys, and all other organs. And as a result, there is polyneuropathy and myopathy. So it can affect the peripheral nervous system, and also like the myocytes, so which leads to cardiovascular disorders. The current treatment options are patisiran, which is on patro. Most of the industry is actually very familiar with on patro. When it comes to lipid nanoparticles, pioneering technology in here, and then inotersen. Yeah. So. So what we have done is actually we have thought about, again, choosing those models. And first I will talk to you about the TTR using Cas9 mRNA. We have used the Ignite as the model system, and the LNP formulations were done using this Ignite, right? And I have given the normal instrumental parameters and the formulation parameters. They are not, they are most of our customers are familiar with this, the parameters that we have used. The lipid composition is the ionizable lipid, DSPC, cholesterol, PEG, TMG, and the ionizable lipid is varied in here, and we use a PNI formulation buffer, and the payload is TTR gRNA with the Cas9 at 1:1 weight by weight ratio, and the typical downstream process is being applied here. And the dilution and storage, we use a PNI cryobuffer and stored at -80°C. So during the mixing, like lipids are in ethanol, and the nucleic acids are in the low pH buffer that I referred to it, and the rapid mixing using the nanoassembler mixing technology. This is NxGen uses invariant mixing. So what does it mean is that lipid nanoparticles remain the same at each time point, and you get a controlled, optimized lipid nanoparticle at the end. So now that we have two different payloads as a drug substance in here, one is Cas9 mRNA, and the second one is gRNA. In this particular case, we thought about doing the co-encapsulation as 1:1. There could be different strategies out there, but we need to, of course, start from somewhere. So we thought, okay, why not we do co-encapsulation and at a 1:1. So sometimes scientists wants to know actually, hey, what do we try to encapsulate? Does it actually reach in there, and does the ratio remain the same? And people could, in that case, use bioanalyzer as a to understand this. So it's just an example data set from the bioanalyzer. And then you could populate into the ratios. And in this case, 50-50 was started with, and almost 50-50 is reached. And this is especially true when you have a high encapsulation efficiency to start with. So when it's a lower encapsulation efficiency situations, it's a good idea to kind of actually go back and check using the bioanalyzer how the ratios are maintained. So ratios and also bioanalyzer can actually provide integrity of the RNA. So it's a good idea in case if the activity was there was a concern in terms of activity. This is one place to kind of look at if it is an mRNA. But if it's a self-amplifying RNA, there are more newer techniques than bioanalyzer, because bioanalyzer wouldn't be able to capture that, and there is capillary electrophoresis and fragment analyzer to understand that. So now, again, we have screened some of our lipids. These are actually preliminary studies that we will be doing more and more lipid screening to this. So the data shows actually that some of our lipids are really, reducing the TTR protein levels. So we have followed a protein reduction in here. That was much more easy to kind of actually follow through as an interread. So that's what we have applied in here. And as I mentioned, TTR guide and Cas9 mRNA is mixed at 1:1 ratio, and preliminary results show more than 75% reduction in TTR protein levels with the tested dose of 3 milligram per kilogram. And we have monitored only at one time point at day 7. Okay, and so the next editing technology we have explored is the base editing, and we have used the PCSK9 is the target. And again, like there is exciting things happening in the industry, and Verve Therapeutics clinical trial is in progress for Verve 101, which is for hypercholesterolemia. And I have put the clinical trial number in here as well. So familial hypercholesterolemia is caused by the mutations of LDLR, PCSK9, and ApoB genes, and the major one is the PCSK9 in here, and this is associated with an autosomal dominant form, and when you have the gain of function variants of the PCSK9, the LDLR levels in the liver is increased, and as a result, it's decreased. As a result, higher LDL cholesterol levels in plasma, which actually causes the problem. So the current treatment options are inclisiran, Leqvio, and Repatha, and Prolent. One is an siRNA treatment, others are actually PCSK9 inhibitors. So we, again, in this particular model, we have co-encapsulated the ABE mRNA, and we have used sgRNA to co-encapsulate in a lipid nanoparticle, and then administered into the mouse and analyzed the blood levels of the protein PCSK9. So a very simple model we have followed. Day 0, the administration, and the day 7, we have looked at the response of the PCSK9 protein. And we could see actually that the lipids have reduced the PCSK9 by around 90%, and using either single dose or double dose, and the dose was at 3 milligram per kilogram. It looked like we could actually probably reduce the dose in the next screens and look at actually more lipids. In this case, mostly one lipid was done, and with another lipid. So again, this is a preliminary results. So far we have talked about in vivo applications, and just to summarize what we have talked about vaccine applications, we have talked about protein replacement application, and we also have talked about 2 gene editing applications for in vivo. So now in this, actually, we wanted to talk about cell therapy application. And in a cell therapy situation, the blood is usually drawn, and then the T cells are isolated, and they are actually genetically engineered with the chimeric antigen receptors directed against CD19 for that particular lymphoma or the liquid cancers. And once it's transduced, it is expanded and put back into the patient, same patient, in a autologous therapy. And Kymriah and Yescarta are the approved drugs for this particular approved cell therapy drugs. The cell is the drug in here. And it's really exciting to see that actually this technology is truly transformational. It changed the life of the patients. And the patient who was in the clinical trial is now 10 years cancer-free and 5 years from the FDA approval. So, I was so excited actually to see these events that's happening. in the industry, and we believe in precision nanosystems that LNPs can also mediate the same job as a viral delivery, and LNP-mediated delivery of nucleic acid therapeutics have the same potential to become similar life-saving genomic medicines. And there is actually certainly lots of advantages to non-viral delivery technologies in terms of regulatory constraints, and also the advantages of doing the work in the clinic itself, like bedside. To start off with, we looked at actually the GFP expression in primary human T cells using proprietary lipids and LNP composition. At that time, we have only MC3, and we have benchmarked our lipids against MC3, and we could see that actually the lipids are performing really well. And the transfection efficiency is like 90% or so. And there are lots of lipids that's really active and also have a high tolerability, which is also very important when it comes to, either cell therapy or in vivo applications. So one thing when it comes to lipid nanoparticles that we have to think about is ionizable lipid is one thing, and we should also think about LNP composition as a drug product, or the LNP that will actually affect the activity a lot. The different components actually plays a role, and for each tissue type, you might actually be surprised to see that there will be favorites for each cell type or each in vivo applications. So in this relevant model, we have used the chimeric antigen receptor CAR construct. And I will not be talking more details about the CAR construct design, but you could see the transfection efficiency, and you could see that it's around more than 80%. And this is benchmarked against the leading electroporation non-viral delivery technology. And there is actually high amount of protein expression in LNPs compared to the electroporation, and also the viability was better for LNPs compared to electroporation. So we have used the normal settings for the electroporation, which is recommended by the manufacturer's protocol in this particular case, because they are two different technologies. In this example, we showcase the dose dependency of a gene knockout, which is the TCR knockout model, using the TCR, sgRNA, and Cas9 mRNA. So earlier we have talked about gene editing in vivo. Now we are actually talking about gene editing in high-value cell types, such as primary T cells, which could actually again become transformative therapies. So the knockout efficiency is showcased in here with respect to the dose. And then you could also see the profile of protein expression changing with respect to the dose. And what the viability is also showcased in here. Then we have again actually showcased a double target knockout to model allogenic cell therapy. In an allogenic situation, the healthy patients, as opposed to a deceased patient, as opposed to a patient who have disease, healthy donors can be taken, and the cells of the healthy donors can be taken and isolated, and the healthy T cells are transformed and then can be put back into the another patient by changing the receptor levels actually, such that there is no host graft diseases happening. So in this case, the TCR knockout and CD52 knockout, and using CRISPR LNPs, and you could see the knockout and viability in here. Then the PNI team have actually explored it further using a multi-step engineering gene workflow, especially for the T cells. And the TCR, first the TCR, it is again to showcase the allogenic model, the TCR knockout, and then the CAR expression. And it is actually further, looked at them. We have further looked at the functional activity by an in vitro killing assay using the sub B15 cells along with the CD19 positive T cells. And you could see actually the specific lysis that's happening really, really working very well with respect to the effector target ratio of 1:1 and 2:1. Further, we have also checked actually how these small scale systems have can be applied in a large scale bioreactor settings, and again whether we can maintain the same level of expression, CAR expression, and the knockouts. And here people have showcased actually the CAR expression and the knockouts and the specific lysis that's happening using the same cell system that we have talked before. And it shows actually that we could really scale it up, the technology. So the exciting thing now is actually, and most of you may be already familiar, we have the off-the-shelf T-cell LNP reagent, and this application note is already available, and people could actually look into it for specific protocols and details. We are first to market this particular off-the-shelf T-cell LNP reagent kit, and it showcases the applications of gene delivery, single gene knockout, double gene knockout, and multiplex technology that I kind of mentioned just in the last message. So in summary, we already kind of know that the genomic medicines have become the reality and the next wave of transformational therapies. And in the last 2 years of pandemic response have helped us to realize what lipid nanoparticles could do, and in terms of a pandemic response, but now the same technology can be applied for genetic medicine advancement. And as I mentioned, every target is now druggable, and as a result, actually, there could be many diseases, especially rare diseases, that do not have a treatment option, can have a treatment option right now. And we could either silence genes, express genes, or edit genes, or repair genes. And we could, using LNP, we could actually do in vivo delivery or ex vivo delivery. And when it comes to ex vivo delivery, we have the option of actually to do it from the patient autologous model or an allogenic model. And lipid nanoparticle is a really flexible technique. And one thing to remember is about the scalable manufacturing. And Precision is a world leading in terms of actually the scalable manufacturing. And most of you are very familiar with our technology again in that side. So with all these, for these all genetic medicine applications, we really need ionizable lipids, and that was kind of coming back to the, the topic of discussion today. We have also showcased the examples in the area, like, some proof of concept in cell therapy area, vaccines, and gene editing applications. Now, if we actually look into the current LNP clinical trials and we try to divide into the modality, you would see this very similar occurrence happening, cell therapy, and gene therapy and vaccines. So in a therapeutic area wise, rare diseases, oncology and infectious diseases, and autoimmune disorders are only 3% right now. So we would work towards actually getting more proof of concepts in the different areas. such that we will have a really good understanding, and then we can help our customers through this process of selection. So in summary, we have a diverse library of ionizable lipids and in-house expertise for targeted applications. And we also have standard off-the-shelf reagent kits to support the early stage research, and also like, to get the familiarity with it, and also like if needed, it can be actually taken into the clinic as well, with more scalable, with more process development and other applications. So one thing to realize here is that the various lipid components influence the nucleic acid protection, formulation stability, target delivery, and drug release. So as an important part of the process is to systematically screen the LNP candidates and assess the biological properties and LNP characteristics with respect to the target product profile. So this is one thing to keep in mind, because most often people always look at luciferase activity in vitro and in vivo, and I would say that, that can be just a start only, but, do not assume that actually that will be the same lead candidates that you are going to get for your specific TPP. So a little bit about the challenges and the complexity of genetic medicines. What are the challenges in here? One, we talked about already, the nucleic acid delivery challenges, and then the drug. There's multiple components that has to self-assemble in an optimized manner to produce the desired product. So basically the structure of ionizable lipids and each constituents and the composition matters, and, and you need some kind of expertise to kind of walk you through that, the selection process. And there is also a lot of knowledge gap. It's an emerging technology, but not that many people in the world are actually capable of handling this. And again, amazing to see the vaccine candidates developed and pandemic response for the pandemic response. But with respect to other genetic medicines, there isn't a huge knowledge out there. So, and also one thing to keep in mind is the different needs for different programs, and sometimes if you have developed lipids or LNPs for one particular application, might not exactly translate to the other application. The design space is very huge. For example, like there is a lot of ionizable lipids to choose from, and there is all 4 components minimal in an LNP. So lots of permutations and combinations and possible, and this along with the target product requirement, its dosing and the route of delivery and administration frequency, it just opens up a huge design space. And as a result, it's good to have some kind of expertise actually being taken from lead experts in this field. Precision always have this one thing in mind. We wanted to provide end-to-end manufacturing of the genomic medicines. Now, Precision is being part of Danaher. So as a result, actually, we have a greater commitment to our customers, and we could actually provide you fit for purpose lipid formulations to the scalability, as to the scalable formulations, and now fill finish and the drug product capability. So we offers actually a wide range of portfolio now, like provide LNP expertise, and the different partners such as ITIVA also plays a role in here, like in terms of the downstream processing. So again, almost same message in here, complete. So we could provide you the complete solutions that translate through the development from early preclinical to the commercial support up to phase 1, phase 2. And And these are the different instrumentations. If in case you are not familiar with, the Spark is actually in microliter quantities. Ignite is the most, most utilized in bench scale quantities. Then Ignite Plus is the newest one, and which gives you more, more opportunities. And then Blaze and the GMP continuous system. So we have a contract services, preclinical services, and the clinical services. We could walk you through the, the process development and technology transfer, and we could also provide you the chemistry, manufacturing controls, and support. And we have that expertise to help you. So, during the pandemic, we have received strategic innovation funding for developing a manufacturing center, and this is going to be open in 2023. This will act as one of Canada's pandemic preparedness center, and this facility is also available for client drug product manufacturing. This facility has clean rooms. So again, very excited about this, and we could offer you more support in the future. So we have already talked to you about ionizable lipids, the different aspects of proof of concepts for cell therapy, vaccines, gene editing, or gene replacement, or and other applications. We are actually working on it. We will bring it to you. And what we wanted to talk about here is actually, together with you as partners, actually we are accelerating the genomic medicines, either in the field of gene vaccines, therapy, gene therapy, and cell therapy. And together, we could provide more genomic medicines. With that, actually, I'd like to conclude my talk, and these are the teams that are involved in this work, mainly Nikita Jain, who was the inventor in the lipids, and then this is the whole delivery team, and we also have help from RNA services team, who provided us with the self-amplifying RNA, and then clinical manufacturing team. Some of the work that you have seen is from the engineer, the scalable, the scaled up formulations, and so hence clinical manufacturing team have provided that. For example, the NHP study results is from the scaled up batch, the process development batch as well. And then the product development team, who is working significantly on the reagent portfolio. And then I would also like to thank Andy Gial, the previous Chief Scientific Officer, and the project manager for the vaccine program. And again, the most importantly, Government of Canada Strategic Innovation Fund and IRAP Canada, and our collaborators, Anna Blackney, Robin Shattock, and team Imperial College London, and Yvonne Perry and team at University of Strathclyde. So, and if you have got any questions, I would be happy to take this at this moment. Yeah.